I'm going to explain the process of gastrula formation in this session. As I mentioned in the earlier session, the blastula results into a stage called gastrula. See, in blastula, as we saw earlier, there's one single layer of cells. Enclosing a cavity called blastocele, and these cells will be blastomeres or blastoderm, the layer would be called as. This blastula would be giving rise to a gastrula, and that process is called as gastrulation. Gastrulation. See, how many layers of cells will be there in gastrula would depend upon what kind of animal it's going to develop into. Suppose if you talk about hydra, that is a cylindrate, then this would have only two germ layers, ectoderm and endoderm. But if it's going to develop into maybe a bird or maybe a reptile or any other triploblastic animal, then this will have ectoderm, mesoderm, endoderm, all three germ layers will be there. So this will be a triploblastic stage in that case. That means one single layered structure would be developing into either two or three layered gastrula stage. That process is called as gastrulation. Now, in this gastrulation process, the layer has to extend in length, the number of cells have to be added to it. So more number of cells, longer a layer, all that has to happen, which involves a number of ways in which this can happen. Elongation of the layer, extension of the layer, or increase in the number of cells, all that can happen in number of ways. Let's know what all ways, what all processes are involved in gastrulation. There's something called as epiboly. Epiboly ideally would mean the layer increases in length. Okay, that is, cells multiply and add to the length of the layer. That's called as epiboly. How will that happen? Suppose if this is the normal layer, then it may start turning in. This layer would be additionally formed. This is called as epiboly. It is found in zebrafish. Process of gastrulation involving epiboly is found in zebrafish. Other ways could be invagination. Invagination, simple meaning, this is the sphere, blastula. This layer starts kind of invaginating. A depression is formed. This invagination deepens further and may result in two or more than two layers. That is invagination process, found commonly in sea urchin. Even Amphioxus might show similar process. Invagination would be the method of modification of cells so as to give rise to gastrula. There's something called as ingration. This is a layer of cells. The cells may get released from here and would give rise to one more layer inside. That's called as ingration. This is found in fruit fly Drosophila. Ingration. And there's something called as delamination. Lamina means layer. So existing layer forms, see, one more layer inside it by multiplication, and this layer gets detached from the existing layer. So lamina as a seed means layer. One layer is detached from the existing layer, resulting into formation of two distinct layers. That's called as delamination. One more way of gastrulation, method of gastrulation found in birds and mammals. There's one more way in which the layer can elongate. It's called as intercalation. What exactly does that mean? See, this is one layer of cells. Another layer of cells. They reorganize themselves such that this is the upper layer, lower layer. The cells have been modified in size, shape. Now they acquire their rotating shape. This is how the layer can elongate. This process is called as intercalation. Then there is something called as convergent extension. Convergent extension. Wherein suppose if there are four layers, they would transform themselves into such that these cells have an increase in length. Further, that would continue and result into formation of longer layer. But instead of four, there are only two. So these have converged into the existing two lines. It's called as convergent extension. That's one more way in which gastrulation can take place. Now that's a broad outline of the various ways in which gastrulation can happen. Let's take specific examples and understand. To begin with, Amphioxus. This is the blastula stage. One single layer of cells. It undergoes invagination. So the next stage would be seen as now remember here this layer is Blastoderm, the cavity is Blastocele. Same thing is continued here. The invagination goes on. And finally, this is what it results into. Here, let's name the parts. The new cavity that's developed, it's called as Archenteron. The opening, it's called as Blastopore. This becomes the upper layer of covering the blastopore. This is the lower layer covering the blastopore. Now, when you talk about blastopore, there's something that we must understand here. This blastopore is going to be a future pore of the gut when the embryo develops from here. It would have an alimentary tract, a gut. This would be one of the openings of the gut. If it leads to opening of mouth in the resulting embryo, then such animals are called as protostomes. Proto means primitive. Stoma means mouth, anterior opening of alimentary tract. Then they are called as protostomes. And if it develops into anus, the posterior aperture of the gut, then such animals are called as deuterostomes. Deuterostomes. The protochordates come under this category, while annelids come under protostomes. That depends upon what the blastopore develops into. As you can see, the blastocele is reducing in size, and finally that blastocele will disappear. This layer would deepen further, and only 2 layers of cells, or maybe 3 layers of cells, whatever, and a cavity. That's nothing but cavity of the gastrula, Archenteron. This is what you see in Amphioxus. Whereas if we start talking about what happens in frog, first thing that we must keep in mind, in frog, eccentrically situated blastocele, these are micromeres, these are megameres. This is the blastula stage. Now, in this blastula, what happens in the next phase? A small involution starts from here. The layer has started involuting. Rest of the cells will remain as they are. Megameres would be seen here. This involution will continue to develop further. And when it develops further, it starts developing a cavity here. This was the earlier blastocele. These are all micromeres. This cavity, which is newly appearing here, is now called as the archenteron. And this layer which is formed here, this is called as the dorsal lip of Blastopore. Same thing would happen on the lower side, and gradually you come across a stage wherein this is the dorsal lip, this becomes the ventral lip, and here is something that slightly extends out and becomes the yolk plug. So here the gastrulation is by involution. The layer kind of grows inside, develops inside. That happens in frog, or let's say an amphibian. Whereas in the case of birds, that's a different way in which this can happen. Let's understand the gastrulation in chick. First thing that we must remember is this egg contains large amount of yolk. So megalecythal egg would be there, and it is telolecithal. So one part would be occupied by the yolk, rest of the part would be occupied by cytoplasm and nucleus. So even after fertilization, it would be seen that the embryo development takes place only in that part where there is cytoplasm. So suppose if this is the entire zygote, major amount is occupied by the yolk, and this is where the embryo development is going to happen. So the blastula would be this is it. Here is the blastoderm. This is blastocele. So whatever embryo development can we talk about, it's going to happen only in this part. Okay. And there, initially, the cells from here would start kind of involuting. So it would be this kind of involution here. gradually this involution will deepen. So if we consider only this part, this is how it would continue. Finally, this thing continues here. So involution would be in this region. Another involution would start from here. So this outer layer, in this case, this outer layer, would become the epiblast. Epiblast is going to become the ectoderm later on. And the lower layer that is developing here, this becomes hypoblast. That would become the endoderm later on. And there's a small kind of elevation here. That is actually a primitive streak. So embryo development would happen only in this region. And below this there is a small cavity. Below this there's a small cavity, which is nothing but a subdermal space, subdermal cavity. That is the blastoderm should be developing above it. Now mesoderm, some cells would start detaching from here and would be forming one more layer in this position. This would become the mesoderm. So this would become ectoderm, this would become endoderm, this would become mesoderm. And there is a small region here which is going to be the prospective gut. Prospective gut. And in the primitive streak, or let us see, this is going to be the upper view from here. This is the primitive streak initially. So here there would be a kind of modification of tissues. This would become the neural fold. Neural fold. So this is going to develop into the nervous system later on. That's how the embryo could be seen in the peripheral view. That is how it could be seen in the sectional view. That is in chick embryo. Gastrulation in chick. So there are various ways, various methods in which the gastrula would be developing. And after gastrula, of course, there will be a process of organogenesis. Organogenesis means different tissues, different structures would keep modifying into different organs. Ectoderm developing into maybe the epidermis, maybe nervous system and certain outermost structures. Mesoderm would develop into musculature, the heart, liver, pancreas. Endoderm would develop into the entire alimentary tract, the connective tissues and so on. This would become the process of organogenesis after the gastrula stage. Do like the videos, subscribe to them, share amongst your friends and family. Don't forget to write your comments, suggestions, queries, etc. Thank you.